Good afternoon, everyone. Really appreciate everyone attending this Journal Club webinar today for American Thoracic Society. We have the allergy, immunology, and inflammation, um, assembly. I'm Miles Hagner. That's going to be moderating this with Ahmed Berry. I'm an instructor at the University of Iowa in the Pulmonary and Critical Care Division. I'm really excited to be moderating this today because I also do work with sarcoidosis, and it's a big passion of mine. And then my co-host, Amit Berry, he is also an instructor with pulmonary critical care at Washington University School of Medicine. And so today we're going to be talking about the paper titled Recruitment of CXCR4 positive type 1 innate lymphoid cells distinguishes sarcoidosis from other skin granulomatous diseases. So it's recently published in JCI, or Journal of Clinical Investigation, in about September of 2024. And so we're really excited to be joined today by the senior author, Dr. Leung. He's an associate professor of dermatology at the University of Pennsylvania, where he directs the Skin Translation Core at the NIH-funded Skin Biology Disease Resource Center, and leads the research residency track at University of Pennsylvania's dermatology program. There he's a physician scientist, where he runs a lab that focuses on how skin and other organs respond to injury and inflammation, aimed at leveraging those molecular mechanisms involved to develop biomarkers to improve diagnosis, as well as identify new treatment options, particularly for dermatologic diseases. And then I have two expert discussants with us today as well. First, Dr. Wonder Drake. She's a professor of medicine within Division of Infectious Diseases at the University of Maryland. She serves there as the Senior Associate Dean for Faculty Affairs and directs the University of Maryland Sarcoid Center of Excellence. She's an expert in sarcoidosis pathogenesis. She's studied it for greater than 25 years, utilizing quite a spectrum of basic and translational approaches, from murine models to sort of human subject research with PBMCs and BAL samples. And then her unique infectious disease expertise in the field of sarcoid has really propelled a lot of the research with involving infectious antigens, like being the first to describe mycobacterial sequences found in the tissue of people with sarcoid, and also that those exposures from that antigen sort of work on the adaptive immune system. And then second, we have Dr. Stokes Peebles. He's a professor of medicine within the divisions of both allergy and immunology and pulmonary and critical care at Vanderbilt. He serves there as the Elizabeth and John Murray Chair of Medicine and is the program director of the Allergy and Immunology Fellowship. He focuses on allergen-mediated and viral-induced lung inflammation, primarily interested in how group 2 innate lymphoid cells initiate, as well as how prostaglandin I2 and glucagon-like peptide 1 receptor agonists then mediate inflammatory responses. And through this research, he's established himself pretty prominently as an expert in working with and studying ILCs. So just a few technical things, housekeeping. So we're going to take all the questions at the end of the discussion after Dr. Leung finishes. If you have any questions, please put them in the chat, and Dr. Berry will read them aloud afterwards. And then if there's any questions after that, then we'll leave it up to the discussion about unmuting and other discussions. Okay. And with that, please take it away, Dr. Leung.
Well, thanks so much, Miles, for that really kind introduction. And thanks to Dr. Drake and Dr. Peebles for participating in this journal club. I'm very excited to hear their thoughts and to have this interaction with everyone in this room. This is a photograph of Philadelphia. So I've been at the University of Pennsylvania for roughly 10 years. And like Miles said, I'm a physician scientist. I did most of my training at Stanford and at UCLA and Caltech, where I trained as an immunologist with David Baltimore as a graduate student. And then I went back to do my dermatology residency at Stanford, where I trained with Sung Kim, who's a diabetologist, so a dermatologist in an endocrinology lab for a couple of years. And then 10 years ago, I started at Penn. And so like Miles says, my lab really studies two main questions. The first is what we're going to talk about today on human skin diseases. So skin is so accessible to the outside world that we take advantage of being able to biopsy diseased skin and unaffected skin from the same patient and use these samples to perform genomics like spatial or single cell RNA sequencing. And from those sort of bigger data sets, develop new hypotheses that we then try to validate with either in vivo or in vitro experiments with the ultimate goal of trying to provide personalized medicine and try and use skin, take advantage of skin being accessible to the outside world. And then the second half of my lab is on wound healing, probably something that we're better known for because we've been studying it for it longer. Basically, if you punch a hole in a mouse's ear, it normally typically heals with a scar and stays open, much like a human earring hole. 10 years ago, when I started my lab, we found some strains of mice where you punch the same hole, the hole completely closed, and the regenerated tissue lacks a scar. 10 years ago, I could do it less than 1% of the time. Now we can do it at roughly about 90% efficiency. And so it's a different type of work, but the second big part of my lab. So we work on two different things. And so here today, we're going to talk about discovering new biomarkers and treatments for sarcoidosis. This is a collaboration with a very close clinical colleague, Misha Rosenbach, who I could never have done this work without his input and help. He is obviously a noted skin sarcoidosis expert in the world. And then Satish Satya was the postdoctoral fellow / senior research investigator that drove this work. And this was a photo of us in Baltimore for the ASOG meeting where I met Dr. Drake last year. And so we published this, it was just recently published in JCI in 2024. And so just briefly for the room, sarcoidosis is a systemic inflammatory disease characterized by organized immune cell aggregates that can be deposited in any tissue called granulomas. And because it can be deposited in any tissue, lung actually ends up being the number one affected organ in sarcoidosis with up to 70 to 75% of all patients. But interestingly enough, skin is the second most commonly affected organ at 30%. Adults get this disease, more common in women and African American, and it's considered an orphan disease because of roughly about 175,000 new cases per year. First-line treatment for sarcoidosis still remains corticosteroids, just as a broad immunosuppressant. Most patients have to be on for at least for about a year, which causes a lot of unavoidable steroid-related side effects like diabetes, osteoporosis, spontaneous vertebral fractures. So really the major unmet clinical needs for sarcoidosis, number one, a lab test to help with diagnosis or measuring disease activity, because this is still for clinicians, this is a diagnosis that we make as a diagnosis of exclusion. You have to remove all the other possibilities before you can give someone a diagnosis of sarcoidosis, which takes time and a lot of effort. And if we could find a lab test that's specifically diagnosed sarcoidosis, that would help these patients significantly. And the second major unmet need for sarcoidosis is obviously instead of having to use a blunt instrument like steroids, hopefully we can find safer and more targeted therapies. And so I always leave this slide just to remind myself that sarcoid within dermatology is often called the great mimicker because it can show up in many different morphologies and locations. So unlike all other dermatology diseases where we make a lot of diagnosis based on distribution and morphology of the skin lesion, sarcoidosis is one of those answers where you can give, if you're ever stuck in grand rounds and they force you to pin yourself on a diagnosis, I'll always throw out sarcoidosis as one of my possibilities. And so it can show up as little red papules on the back, a large nodule, obviously on the hand here. Probably the most common presentation of sarcoidosis in skin are these nodules around the nose and nasolabial folds. But you can also have this hypopigmented variant or even this serpiginous variant as well. So it's a clinically heterogeneous disease that is difficult to diagnose. So the current model for how granulomas are formed, so this is really just the basics here, is that there's even though sarcoidosis was discovered more than 100 years ago, it's an unknown inciting agent, whether it's infectious or not, up to debate. Antigen-presenting cells, whether they're macrophages or dendritic cells, are activated by this agent, and then they talk to the T cells to form these organized aggregates of immune cells or granulomas within different tissues. And macrophages form epithelioid cells, become epithelioid cells or multinuclear giant cells. And then these macrophages in the center are then surrounded by peripheral T cells. And then the major cytokines that are released within the granulomas are interferon gamma and TNF alpha. But many people in the field have identified other these blue colored cells that are at the periphery of the granulomas, including Dr. Drake identifying Th17.1. People have shown B cells are present too. And so how these additional immune cell types might play a role in granuloma formation, largely still not as well characterized as we hoped. And so we took this as an opportunity to perhaps identify additional blue cells that people may have overlooked, or to maybe figure out how some of these mechanisms are being regulated to form these sarcoid granulomas. And so, like I mentioned in my first slide, we take advantage. I'm a dermatologist. Skin is on the outside of the body. It's very easy, accessible tissue to biopsy compared to lung and heart and liver or internal organs. So, for example, just to put this like in a bigger perspective, at least I'm unaware of. I don't think there are any publicly available data sets of pulmonary tissue sarcoidosis available online. No one's done single cell RNA sequencing of lung tissue itself. People have done it on BAL or bronchoalveolar lavage samples, but they haven't done anything on tissue. There's only one single study on single cell RNA sequencing of cardiac sarcoidosis tissue. And so we thought maybe there's an opportunity for us to study some mechanisms of granuloma formation, mechanisms of systemic disease by looking at the skin. And so we collected affected and unaffected skin, and as well as blood from all of our granulomatous patients. We did basically a couple of techniques. We did spatial transcriptomics, we did single cell RNA sequencing, and we did some flow cytometry. And from this rather large data set with an integrated analysis, then we drove some hypotheses, and we followed up with some mouse models of granuloma formation, as well as some in vitro assays. And so I hope to lead you through this data set as we speak. So for our single cell RNA sequencing data set, we collected affected and unaffected skin from 28 different patients, 18 with sarcoidosis and 10 with non-sarcoidosis granulomas. And these are some of the diagnosis of the non-sarcoidosis skin granulomas that we took. And we use this really as our control group. And so from these 28 patients, we're able to have 55 biopsies. One of the control samples did not work. And we're able to generate a single cell RNA sequencing data set of over almost 500,000 cells, which is really large. And then we basically use very standard methods to annotate and cluster our cells. And I'm just in interest of time, I'm going to skip those. But if anyone wants to talk about them, I'm very happy to go into great depths of how we annotated our cells. So one of the first things that single cell RNA sequencing offers you is an unbiased catalog of what cell types are you looking at in your samples. And so this is a rather complicated graph, so I'll walk you through it. We have 4 columns. The purple, it's basically the purple and the blue are unaffected skin samples from either non-sarcoidosis skin granuloma patients or sarcoidosis skin granuloma patients. And then the green and the salmon color are the affected skin of non-sarcoidosis as well as sarcoidosis patients. And we're looking on the x-axis. And so we had identified 2 different macrophage populations within our single cell RNA sequencing, a little bit like M1 versus M2 or inflammatory versus anti-inflammatory macrophages. And you can see in the affected skin samples, higher recruitment of those macrophages compared to the unaffected skin in both populations, things that we would have expected. We also see more recruitment of T cells. People have known this for years. So Th1, including Th17 and Treg cells. But what we were looking for were perhaps we were trying to find an immune cell type that is unique to sarcoidosis that we don't see as highly elevated in non-sarcoidosis skin granulomas, and we centered on innate lymphoid cells, or ILCs, which were more recently identified, and these are the innate counterparts of T cells. And you can see here that skin sarcoidosis patients had higher levels of ILCs compared to non-sarcoid skin granulomatous patients. And so we conclude that sarcoidosis skin uniquely recruits ILCs. Now, I mentioned that ILCs are rare and difficult to track. And so we want to make sure that this result that we're basing this whole paper on was actually correct. And so we verified this by uh, 4 different methods. And I hope to convince you by the end of this, that we're able to really identify single cell ILCs within our single cell RNA sequencing database. And so for the first thing we did was we adopted a flow cytometry gating scheme that was published by other people people have used to isolate different types of ILCs, ILC 1, 2, 3, and as well as NK cells. So we adopted a published flow cytometry grading system. For our next 7 patients with granulomatous skin diseases, we actually took their skin, we made them into single cell populations, and basically used flow cytometry to analyze how many ILCs do I see in my skin biopsy samples. And so you can see here for 4 sarcoidosis patients and 3 non-sarcoidosis skin patients that we see that sarcoidosis patients had elevated numbers of ILC type 1 in their skin, and we did not see any in any non-sarcoid skin granuloma patients, as well as the unaffected skin, really confirming what we saw with single cell RNA sequencing, but using the gold standard. The second thing we did was, because we could now flow purify these ILCs using flow cytometry, we did bulk RNA-seq on them. And so bulk RNA-seq obviously gives us a much deeper and richer gene expression data set compared to single cell RNA sequencing. So we took the bulk RNA sequencing genes that we saw that were upregulated in those cells, and we compared them to our single cell RNA sequencing cluster of cells that we identified within our single cell RNA sequencing database for ILCs. And we found that 72% of the genes that we found in our single cell cluster were present in our bulk RNA-seq database, including 8 out of the top 10 genes, really adding more confidence that what we have identified in our single cell database were indeed ILCs. And then we did 2 other ways. I'm going to go very quickly now. The third way is there's an ImGen database. Basically, people have phenotyped mouse immune cells very, very well. And so you can just upload your candidate gene marker list for your gene expression list into the database, and it'll return what its cell types it most closely matched. And I understand for our human ILCs, it returned a mouse ILCs as the closest immune cell type. And then finally, people have published flow-sorted, purified populations of ILC 1, 2, and 3 and other papers. And so we just downloaded their gene list and compared it to our gene list. And here I'm just trying to show you that ILCs seem to match the flow-sorted ILC. Our ILC population seem to match the flow-sorted ILC population the best when you just eyeball gene expression changes. So we think from those 4 different methods that we've really identified ILCs within our single cell database, and that they're elevated in sarcoidosis skin. Okay, so ILCs, what are they? These, I just mentioned them briefly. These are the innate lymphoid cells, or the innate counterparts of T cells. And so there's 4 different flavors: ILC 1, 2, 3, and NK cells. And these all mimic different versions of T cells. So ILC 1 mimics Th1, ILC 2 Th2, ILC 3 Th17, and then NK cells mimic CD8 T cells. And when I say mimic, they actually just share the same effector cytokines. And when we first got this result, it sort of made sense to me, because we know that Th1 CD4 cells play an important role in sarcoidosis. We know that interferon gamma and TNF are the most important cytokines involved in granuloma formation. So why wouldn't ILC1 potentially play a role in granuloma formation when it makes very similar cytokines? And so we want to ask next, where are these ILC1s that we're seeing in our sarcoidosis skin? Are they actually located within the granulomas themselves, or are they just innocent bystanders and just hanging out in the rest of the tissue that that isn't being actively involved? And so here we did spatial transcriptomics, and this was 3-4 years ago. So we used just a regular Visium platform, and this is the H&E of a non-sarcoidosis skin sample. And so this is the outside layer of the skin, and this is the inside layer of the skin, sort of dermis. And you can see the dark purple here is where the granulomas are. And then it's the same thing for a sarcoidosis granulomatous patient, where you can see the dark purple areas are where the granulomas are. And so spatial transcriptomics gives you gene expression within each of the dots for Visium. And then from the different gene expression profiles, you can then extrapolate what cell type they're that might be residing there by assigning identities based on marker gene lists again. And so when we call for ILCs or ILC-specific marker genes and where they might be located within these samples, you can see for the sarcoidosis patient that this orange color is where the program had picked up ILCs potentially being present. And these ILCs actually correlate to where these purple granulomas are. Meanwhile, we don't see much orange in the non-sarcoidosis skin sample. Really, again, another just a line of evidence saying that perhaps ILCs are playing a role within sarcoidosis granulomas. That said, Visium has a 55 micron spot size, which is really 2-3 cell diameter. This was in Visium HD. And so we want to follow this up by using histology. And so we did just traditional histology for ILCs now. We've done this for both lung and skin sarcoidosis granulomas. In the interest of time, I'm only showing you the lung results, but this is important to us because we were wondering whether what we're finding in skin granulomas can actually apply to granulomas in other organs caused by sarcoidosis. And so this was an important thing for us to do. And so here we're performing ILC histology. Dr. Peebles can tell me that this is not easy to do. There's a huge lineage negative channel that you have to incorporate with the histology. So we did that. So the lineage negative channel has marker genes for all the other immune cell types that you could think of would appear in the lung or in the skin. We use 2 marker genes for ILC1 specifically, either ILC7R or TBET as marker genes. And so when we did, if you merge this histology, you can see that the red only cells would be lineage negative IL7R positive or lineage negative TBET positive. And you can see all these red cells were really at the periphery of this granuloma here, really suggesting to us that these were the blue cells that people had. These might be some of the blue cells that might reside at the periphery of the granuloma, and perhaps they might play a role in granuloma formation. Okay, and then so next, we, in addition to collecting skin, we collected some blood from these patients because we had gone through the trouble of doing a flow cytometry panel to analyze ILC. We decided to use these blood samples as well on our flow cytometry panel. And so when we did flow cytometry on all of our blood samples from these 28 patients, you can see that sarcoidosis granuloma patients had elevated levels of ILC1 compared to HV, which is healthy volunteer, compared to G, which is non-sarcoidosis granuloma patients. And they did not have any elevation of either ILC2, 3, or NK cells. I just don't know why that data got lost while I was making the slides. So in conclusion, sarcoidosis patients had an 8-fold increase in circulating ILC1s in their blood. When we looked at this data, we were really struck by the dichotomy of seemed to be a high population of ILCs as well as a low population of ILCs. And so when we subgrouped our patients based on whether they were treatment naive versus getting active treatment versus healthy volunteers, very nicely for us, we saw that the high population correlated with naive, non-treated patients compared to the lower population that correlated with the treated patients. And this single patient that still had high levels of ILC1s actually was not responding well to their treatment regimen. And so this data suggests to us that circulating ILC1s correlate with treatment status, and perhaps they may represent a nice disease activity biomarker for sarcoidosis. And so now that we've identified that ILC1s are more present in the skin, they're more present in the blood, we want to ask, well, are they really necessary for granuloma formation? So one of the weaknesses of the sarcoidosis field that's well known for the last 20 years is that there just hasn't been a consensus mouse model for sarcoidosis. All the current models all have weaknesses one way or another. And I know there's no perfect model, but there is no consensus model. And so we want to use the best available model to answer this question. And so we screened actually 4 different models, but 3 different models, 3 to 4 different models that form pulmonary granuloma. So these are models using either carbon nanotubes, cadmium nanoparticles, or QDOT, as well as the TSC2 flox-flox-lysine-Cree transgenic mice that was recently discovered by Thomas Weichert. And what we did was that we used a flow cytometry panel, again, adapted a mouse flow cytometry panel now this time for ILC1. And then we asked all these, we induced granuloma formation in all of these mice. And then we asked, do we see ILCs in any of these granulomas? Because if we're going to test if ILCs are necessary for granuloma formation. I hope that ILC1s are present within these granulomas. And here I can show you by flow cytometry, and we narrowed it down to both the QDOT model and the TSC2 flox mice. Both have ILCs in their granulomas, and this is the data showing you here on by flow cytometry in a wild type mouse. Without QDOT, you have no ILCs in their lungs, but in a wild type mouse, after QDOT exposure to induced pulmonary granuloma formation, we find a lot of ILCs. ILC knockout mice exist, but in order to make an ILC knockout mouse, you also have to you have to create this mouse in the background of a RAG2 knockout mouse, which lack B and T cells, but have ILCs and macrophages, so they can still recruit ILCs to their granulomas, to their lungs, but we don't see any recruitment in the ILC knockout mice, which confirming that our flow cytometry panel works, as well as confirming that we can see ILCs in these mice. And so with this data, we're like, okay, we can use this cadmium nanoparticle at least to study granuloma formation that ILC1s typically participate in. And so the experiment we did was we took wild type mice, we took RAG2 knockout mice as a control population. We took these ILC1 knockout mice. We exposed them to QDOT. We waited for 30 days, and then we basically harvested their lungs, did whole mount histology, and asked what happens to the granuloma formation. And so here you can see in a wild type lung, whole mount, a mouse, wild type whole mount mouse lung, you can see this lung is studded with these dark purple aggregates throughout the lung, which are granulomas. Here you can see in the RAG2 knockout mice, they don't form really as distinct purple granulomas, but you can see that the macrophages are still able to aggregate into organized structures, as you can see here. But in the ILC knockout mice, which are RAG2, IL2, R gamma knockout mice, you can see that we don't even get any macrophage aggregation as well. And so no granulomas are forming. And so when we quantified this by just looking at number of granulomas per high power field, we saw fewer granulomas in a RAG2 knockout mouse compared to a wild type mouse, but we saw even fewer granulomas in an ILC knockout mouse compared to our control mice. And so we conclude that sarcoidosis granulomas specifically recruit ILCs, and they seem to be necessary for granuloma formation in mice. Okay, so in addition to immune cell type, single cell RNA sequencing also returns gene expression changes. And we sort of glossed over, I mean, not glossed over, but that's the second major data that single cell data sets are able to give us. And so we want to take advantage of this ability. And so the first thing we asked was, tell us, what are the most induced signaling pathways in sarcoidosis skin compared to non-sarcoidosis skin? Are there any big differences? And so what we have here is all the different cytokines and receptors that are available in the immune system, or for most, not all, but most of them. And this is healthy skin. This is our sarcoidosis skin. This is our unaffected sarcoidosis skin, and then the non-sarcoidosis samples. And then here on top, single cell data sets for other skin inflammatory skin diseases. So atopic dermatitis, psoriasis, vitiligo, bullous pemphigoid, and just to see if sarcoidosis actually induced a signal that we don't see in other diseases. And you can see very easily, this is a dot plot where the size of the dot is tells us the percent of cells that express this receptor or ligand, and the dark and the color talks about average expression compared to unaffected, compared to all the other data sets. And so you can see that the top hit, easily see the top hit was CXCR4. And so CXCR4 is a receptor that works with CXCL12, which is this canonical ligand, and it's a well-known signaling pathway for homing of immune cells. And so first we want to double check, okay, so if cell chat, this is generated by cell chat. If cell chat tells us that CXCR4, CXCL12 seem to be upregulating sarcoidosis, do we really see it by transcript? And so here, just using, again, returning back to our spatial transcriptomic data sets, affected sarcoidosis skin have a lot of induction of CXCR4, which we don't see in unaffected sarcoidosis skin or psoriasis or healthy skin. And in the ligand is CXCL12, where we see robust induction as well. In fact, a little bit even more in psoriasis, really suggesting that what we saw in our single cell data set was confirmed in our spatial transcriptomics data set. We also took it a step further. We confirmed it by histology, so we can see expression of CXCL12 within sarcoidosis granulomas. And these are human sarcoidosis skin granulomas. human sarcoidosis skin granulomas. And so because CXCR4, CXCR12 is a skin homing cytokine pair, we look to see if using an inhibitor for CXCR4, which is already FDA stem cells for some patients called plerixafor, we wondered whether we can block this migration. And so we did a trans-wall migration assay using PBMCs from sarcoidosis patients as well as PBMCs from healthy volunteers. So you put these cells on the top, you put your ligand on the bottom, you wait 3 hours, 4 hours, and then afterwards we collected the cells that had floated to the bottom, did flow cytometry to identify their identity. And you can see here that typically there's a 7-8 fold change in the number of immune cells that respond to CXCL12 and sarcoidosis PBMCs compared to healthy volunteer PBMCs, and that this additional migration ability was blocked by this CXCR4 inhibitor or plerixafor, really suggesting that CXCR4 inhibitor really blocks immune cell migration and may be able to help prevent granuloma formation. And so with that data, we asked, well, can we prevent granuloma formation if we use a CXCR4 and that same CXCR4 inhibitor? So we repeated this mouse model that I spoke to you earlier. Take some wild-type mice, give them Q dot. We also began to treat them with the CXCR4 inhibitor. Again, day 30 days later, collect their lung tissue. And so in our control group that just received PBS and their osmotic pumps, obviously a lot of granuloma formation. In our plerixafor treated mice, we got much fewer granuloma formation, really suggesting that CXCR4 inhibitor could potentially be a targeted therapeutic for sarcoidosis, at least in mice. And so with that, I'm just going to summarize very quickly. So I hope I've convinced you that in non-sarcoidosis skin granulomas versus skin sarcoidosis granulomas, there seem to be different immune cell types that are involved in each of these. There's different immune cell types involved in these different diseases, suggesting that there's a disease-specific granulomas. I'm not sure people had figured that out prior to this work. And for sarcoidosis specifically, though, we had found that some ILC1s, or these blue cells that are at the periphery of the granulomas, are present that we don't typically see in non-sarcoidosis granulomas. The second cell type that's actually specific to sarcoidosis granulomas that didn't have a chance to share the data with you today is actually B cells. We use a mouse model of granuloma formation induced by cadmium nanoparticles, and we're able to prevent this granuloma formation either by deleting ILC1s or by using the CXCR4 inhibitor plerixafor to reduce granuloma formation. So some of the conclusions that I'd like to make today would be that ILC1s are a potential tissue or circulating biomarker for both sarcoidosis diagnosis or to monitor disease activity. CXCR4 inhibitors may be a new potential targeted treatment for this disease. And 3, I hope to convey that we should really try to use skin as a window to study systemic inflammatory diseases. By studying, these processes in disease, human skin, hopefully we can get some insights into what might be happening in other organs as well. And so future directions that are ongoing for this work now, we're testing whether ILC1 is a diagnostic biomarker by measuring ILC1 levels in all of our new patients suspected with either skin or lung sarcoidosis here at Penn. 2, we're measuring, cutaneous sarcoidosis or skin sarcoidosis. Misha Rosenbach has developed a scoring system to clinically measure disease activity. And so we're trying to understand whether the scores that they generate using this skin disease activity measurement correlate with levels of ILC in the skin, in the blood. And so we're just doing a cross-sectional assessment in all of our existing patients. 3, we are actively starting a clinical trial to test plerixafor by repurposing it for treatment of cutaneous sarcoidosis. And then 4, our lab is very interested in trying to understand how ILC1s modulate macrophages to make granulomas. And we think this makes some sense because ILC1s are innate cells. So we think perhaps that ILC1s are actually recruited to the affected lung first, sooner than T cells, and might be able to help jumpstart the process to talking to macrophages by exchanging cytokine interactions. And so with this, I'm going to end. This is my group. We have a wonderful group of people. I mentioned earlier the Satish Sati drove the work. We're funded by both multiple grants from the NIH and VA, which we're very lucky, and as well as some of these other sources. These are our clinical collaborators, including Misha Rosenbach. Thank you so much for your interest in inviting me to present today, and I'd be happy to start our discussion.
Dr. Leung, thank you so much for that incredible presentation. Just a fantastic work that you've used multiple modalities to sort of verify. Really interesting and thought-provoking work. I think we can now open up the conversation to our brilliant discussants. Perhaps Dr. Drake, you could lead us off, and Dr. Peebles, you could give us your thoughts shortly thereafter.
Yeah, so congratulations, Thomas, again on an excellent body of work. I'm very impressed. I enjoyed reading your paper. Read it several times just to make sure I understood. So I was thinking it's a little bit tough when we look at figure 5, where you're looking at healthy volunteers versus sarcoidosis and looking at the percentages of live CD45 cells, and you show this disparity. And then you further subdivide them by treatment and see that the ones with high levels are untreated and the ones with the treated have lower levels. And it's a little bit, because it's a point in time, I wondered if you and Misha had worked together to maybe get patients longitudinally to understand, because it's a couple of ways you could interpret that. Because it's a point in time, it could mean that they're high because they're protective and they're not on medicines that it can ameliorate its capacity to be its migration. Or it could mean that, or the converse could be true, if that makes sense. So I'm wondering, I feel like until we've followed patients longitudinally, like take a Lofgren's patient, for example, who you probably would not see in derm clinic, but a patient who presents with acute sarcoid that you know they will resolve their disease, and follow those levels over time. That's what would distinguish for us clinically, is it protective or not? Does that make sense what I'm...
I love that. So I love that idea. I wrote several grants proposing that idea.
I didn't review it.
and they always got well, none they did not get, anyways, they didn't get funded. And actually the response back was, that's too much work, it's too complicated, you should do a cross-sectional analysis. And so that's why I've now adopted this cross-sectional analysis discussion. Like, this is what I say in public, but I agree with exactly what you said. It's something that we really wanted to do. It's something that I thought was actually very doable. I'm like, look, we see these patients regularly. This is not like, I don't think it's that difficult. And within the realm of reality for at least a 3-year grant. And so, but such a great idea. And I hope you read my grant next time.
Okay.
So I wonder, do you have other questions?
No, I thought we were going back and forth, so I'm sorry.
Okay, well, we can go back and forth. That'd be great. No, that was Thomas. That was a terrific presentation, and you know, it really is a tour de force work. I had some questions, not, I mean, more general questions, I guess, philosophically, about your work. One was, you know, as you mentioned, CXCL12 is the ligand for CXCR4. So what do you think the driving force is for the production of the CXCL12 that's recruiting these CXCR4 expressing ILC1s? And is that a potential target too? Obviously, they're not, you know, they're seemingly around the periphery of the granuloma from the great pictures you showed. But what do you think it is that's driving the recruitment of those ILC2s and I mean, it's not ILC2s, ILC1s, and could that be a therapeutic target, I guess?
I think that's a wonderful question. We haven't figured out what is expressing CXCL12. I always assumed it was the macrophages. So my assumption has always been the macrophages show up. At least we have some mouse model data that the macrophages show up first, okay? And then they're trying to engulf whatever the antigen is. And I think they begin to secrete CXCL12 first, and then their CXCL12, which they can, and we've shown that, that their CXCL12 then starts to recruit CXCR4 positive immune cells, including the innate, including ILC1s.
Okay. And so, okay, so it's a result of the activation of the macrophages related to the antigen, and Dr. Drake would say it's tuberculosis. So yes, right.
I'm
So
for that
another,
too. I'm totally in that camp as well.
right. And I guess the other question I have, CXCR4 retains cells in the bone marrow. So for instance, there's WHIM syndrome, which is where you have you have an excess of CXCR4 and it keeps neutrophils in the bone marrow and they can't get out. And so in this case, I guess the question is, and I get we had a guy in our lab, Matt Steer, who showed that CXCR4 was really important in keeping ILC2s in the bone marrow, and you could get rid of that by treating the cells with the ILC2 progenitors with IL-33, and it downregulated CXCR4, and then therefore they could get out into the periphery. So I guess the question is also in your if you're in your potential studies, if you use a CXCR4 inhibitor, you're going to increase the egress of the ILC progenitors out of the bone marrow. And do you see any potential problems with that?
Yeah, I think that's a great question. I didn't know that. I'm not sure I knew. I did not know that CXCR4. I knew about the neutrophil part and about the bone marrow stem cell part, that for sure. But I didn't know that CXCR4 modulates ILC egress out of the bone marrow, and that's just me not knowing the literature well enough. And so that's a really good point.
Yeah, thank you.
What I've been struck by most of all isn't so I'm not sure that the CXCR4 inhibitor only blocks ILC1. Actually, I think that the CXCR4 inhibitor also blocks the T cells as well as macrophages. And so I think by just dampening but what was surprising to us was that CXCR4 was just so represented within the sarcoidosis granulomas. So I don't think it's an ILC1 specific mechanism. I think it'll be blocking multiple immune cell types, and I would never think that this would be an ILC1 specific mechanism.
Great.
That's great.
And that'll be your next rant. Maybe you can figure that out.
Exactly.
And so I should have wanted to ask my question because Stokes, your question was very close to mine. So in other diseases, these cells do affect gut permeability. The CXCR4 positive cells increase gut permeability. And, we are interested in the role of the gut microbiome in the fibrotic lung disease in marine models and people. And so we're writing up a paper now actually showing bacteria trafficking with these immune cells. And so I guess I wonder, like, I guess I'm not trying to figure out how to ask the question. That's probably why I was hawing a little bit stuck. So I guess I'm wondering, could it be that the enhanced gut permeability you get from CXCR4 positive cells, could they be getting to the lung? And then that's what's, I know you're looking at skin, but I'm thinking about in terms of lung. And that's driving more severe disease also. Because something that I've noticed the field hasn't done, like in granulomatous diseases of known etiology, like TB, histo, people are starting to do microbiome analysis on it. And it's very interesting in that it isn't just the pathogen that we think of. I've always thought of it as the pathogen and immune cells, but there's a whole family there, And so I wonder, could that have a role too, outside of its immune role, the CXCR4 signaling, increasing gut permeability, could we be getting metabolites and pathogens out that are also worsening disease?
I think that's a totally possible and plausible hypothesis. We're not looking at gut microbiome.
Yeah.
People in our field are looking at skin microbiome a lot, but beyond my expertise, but it makes sense to me.
Yeah, that's a safe answer.
Nick Hogan on the chat said, I was wondering, thank you for that wonderful presentation. I was wondering how numerous the ILC1 population is compared to Th1 T cells and granulomas. Are they playing the same role? So ILC1 is like 1/10 or 1/20th of the Th1 T cell population. So it's a very minor cell type. And so that actually, that everyone's like, oh, it's such a minor cell type. How can it be playing such a large role? And that's a valid question and a valid It's something that we've been thinking about. My only explanation there. Okay, so one, our mouse models suggest that ILC1s play a role, but it's in the absence of T cells. And so, but there's no mouse model right now of an ILC1 knockout without knocking out T cells, because it's impossible to make or my understanding is it's impossible to make that mouse for ILC1 specific. There are ILC2 and ILC3 specific knockout mice, but there are no ILC1 specific knockout mice without knocking out T cells. So it's hard for us to test that hypothesis. But the model that I have is perhaps the ILC1 just show up earlier to help jumpstart the process, allowing the granulomas to begin 2-3 days earlier than having to wait for the T cells to show up first, and then maybe supplemented by the T cells when they get there.
Thomas, I'm going to add something in, maybe helps with this. Is if you look at ILC2s, for instance, and I'm not an ILC1 expert, we've done a lot of work with ILC2s. If you look at the IL-5 production of ILC2s, it's 5 to 10 times more than a CD4 Th2 cell. So on a per cell basis. So it's very possible that your ILC1s, I don't know whether you've looked at, MFI of interferon gamma in your ILC1s compared to the CD4 Th1 cells. But I would look at that, because it's very possible that you're going to find a whole lot more interferon gamma being produced on a per cell basis than your CD4 T cells. And that may be, you're talking about jumpstart, that would be a huge jumpstart. And it may be that that by itself may be enough to really get the granulomatous immune response started.
That's amazing. We haven't checked yet, and that's something that we should definitely do.
I was just going to add as well that, so I study epithelial biology with my mentors here, and I think that the epithelia in the airway itself, and probably in the skin, but possibly more in the lung because of the propensity of lung involvement in sarcoidosis, that is like jump starting or augmenting that initial response to ILCs, and then ILCs is then doing it even further. At least that's what I'm sort of hypothesizing, that it's, there are multiple sort of like rate limiting sort of things that just make it sort of wildfire, like after the immune system and things.
It's totally reasonable.
Dr. Leung, I had a question, and maybe Dr. Peebles or Dr. Drake can weigh in as well. Sarcoidosis is obviously a disease of granulomatous inflammation, but one of the things, I'm a transplant pulmonologist, one of the things that I see patients for is end-stage burnout sarcoid with fibrotic lung disease who need lung transplants. So the question I have for you is, you've proposed this very interesting mechanism of utilizing prolixafor, and Dr. Peebles, astutely pointed out that this may release other cells from the bone marrow that could have an impact on inflammation and immune responses. I know this is very early, but what are your opinions, and perhaps the expert discussants can weigh in as well, on whether, abolishing granuloma production would also prevent, downstream fibrosis or scarring later in the disease? Is getting rid of the granuloma itself sufficient, or is it possible that, the mechanisms that have been activated will allow fibrosis to occur even without the actual granuloma forming?
I would love to hear their input. I'll give you my 2 cents, and then I'm going to defer to the real experts. Okay. So if you think about granuloma, or I think of granuloma as just really just chronic inflammation, and the chronic inflammation is what's driving the fibrotic change, the end stage fibrotic changes. So one of the caveats for all these mice models that we use is that none of them actually generate fibrosis, because I think the chronic inflammation doesn't last long enough. It's not like the bleomycin mouse model where like you get tons of fibrosis that we use for idiopathic, that people use to study pulmonary fibrosis, but none of that changes ever show up, right? And so for me, that tells me, oh, maybe it's the granuloma that causes a lot of chronic inflammation, and then that leads with the ultimate fibrotic change that we see at the end stage, and certainly something that we want to study in the skin, obviously for scar formation and my wound healing stuff, and that's how it correlates with me. But that would be my 2 cents. But I defer to the real experts.
So I'm gonna say I only have a little to add there because Stokes, this is his area. One of the things that the field of sarcoid hasn't done, because we haven't been able to, is learn that granulomas are dynamic. So when you look at like Joanne Flynn, her work, she shows very beautifully that, we take a snapshot of granuloma and we see these immune cells present, but they're actually going in and out, transversing, going throughout the body, then coming back and using a monkey model. So I really don't think we understand, because we don't have the tools to do so, that we truly understand the immune dynamics of sarcoid granulomas, and the fact that it may be something other than a single antigen that's driving it. Like when I read their work, it is starting to convince me more and more that there is this inciting event that drives these collections of immune cells, but that event is then heavily ameliorated by other pathogens that may honestly be commensals. They're not even necessarily causing disease, but they're stimulating this potpourri of immune cells. So I think it's hard for us to say in sarcoid, because we don't have the tools to answer that question, like they do with TB, with a monkey model with TB, and you can biopsy over time and show this T cell was once in the granuloma and now it's in the peripheral blood and things like that.
I don't have anything else to add over what Dr. Drake just mentioned.
I was also going to say, I think what you were sort of pointing out there, Wonder, was that I also think that's why occasionally, but not as commonly, it would like TB and histo, occasionally we'll see sarcoid granulomas that are necrotic, but they're not often. But it's like, I think when symptoms initially come on, they get evaluated, seen. It's not, that level of inflammation hasn't been persistent for long enough to develop some of that necrosis.
Mhm.
But I at least that's what I think could be the cause of why we find it sometimes, but much less often than TB. I'm not sure if you would think that that also explains it, but because I know we
I think that's
tend to think of it
definitely
as non-necrotizing, but it can be sometimes.
It can. I mean, it's about 3%, right? In necrotizing sarcoidosis granulomatosis. Yeah.
Do you guys think that infectious granulomas and non-infectious granulomas use the same mechanisms to generate the granuloma structure itself, or do you think that they're totally, completely different processes?
I personally think there can be immune overlap, but it doesn't have to be.
Yeah.
As I've been studying um, you know, I've been studying more Joanne Flynn and what she teaches us about TB granulomas to try to apply it to sarcoid, because you see immune responses to mycobacterial antigens in sarcoidosis. But we honestly, I mean, this is what's really hampered the field. And this is why I started using the Bleo model, because there is some immune and pathologic overlap with sarcoid, just to try to learn something, because it significantly curtails the questions we can answer.
Yeah.
I was going to ask if no one else had a burning question. I was looking at some of the supplementary figures in the paper and talking about, you initially mentioned when with the biopsy and skin sampling and the characteristics between the cohort of patients with sarcoidosis. Looking at sort of the disease involvement that you guys looked at, it I feel like race and ethnicity was fairly balanced, but like you mentioned at the beginning, lung having the highest involvement, you only had, I think, about 40% or 30% sort of lung involvement. And I was curious, is that, do you think it's just a component of like the dermatology clinic and sampling bias, or is that like possibly more representative of like Philadelphia and the area that you treat? And then secondarily, did you happen to look at the differences in the different pathways and the expression of the ILC1s across disease involvement, and if it differed?
Okay, so part one answer is, my guess is that we collected all these samples from a skin sarcoidosis clinic, and so I'm sure we're going to have overrepresented patients, and then people with lung involvement probably don't have to come to the skin sarcoidosis clinic because they're getting much stronger treatment and their skin disease improves. So I think that might explain the difference in the 70% versus 40%. Your second question is a really good point that I don't think we want to. So That's just disease severity, and do the ILC numbers seem to correlate with disease severity is another way to maybe potentially tackle that. And our sample size was too small. Like we just didn't have, like, it's really hard for we didn't have a good way to grade activity in the lung or in cardiac very, very well. We're starting to talk to our pulmonary colleagues as well as our cardiac colleagues, but we haven't had it just has been harder for us to quantify activity. Serial CT scans, things like that, but we haven't done it yet.
And then I think just one final point to that, if I could, before kind of wrapping up here. Would that spring off that same question, since the CXCR4 and the CXCL12 sort of axis, predominantly you were talking about how trafficking, would you think that would suggest there'd be more extrapulmonary involvement than maybe we observe, just in the sense that would maybe be having immune traffic to different, because even in the unaffected areas you had some tissue that still had positivity for that. And I was just thinking, I mean, we do have 30 to 40% or so of extrapulmonary sarcoid in the population, but I was thinking if this is such a strong driver of the disease, maybe you'd have even more. But I'm not sure. Does that make sense?
I'm not so sure. I don't know is my answer. I think if CXCR4, CXCR12, well-established skin homing axis, no doubt. I think it should work in any organ. And so I think it's really where the macrophages that are secreting the CXCL12 are being activated first. That might dictate the extrapulmonary involvement, perhaps. I don't know. It's too early. I don't have any data to support any. I'm just pure speculation. Verbal diarrhea.
Gotcha. Well, thank you so much.
This is
I hope we can
that's
chat
the
about it at WASA.
Yeah, just keep doing what you're doing. I mean, this is excellent. We're learning,
Thank you so much.
Yeah.
Thanks, you guys. I appreciate it.
Yeah. So with that, just want to thank everybody, Dr. Leung, for an amazing presentation. That was so cool to go through the various things that you were able to accomplish in that paper. So thank you. And then the discussants, I think Dr. Peebles had to possibly go, and then Dr. Drake, thank you so much. And Dr. Berry for helping me co-host. Appreciate everyone joining.
Thanks for an invitation. Thanks for everyone for coming. And thanks, Dr. Drake, for coming too and lending us your expertise. And thanks to Dr. Peebles. I appreciate everyone.
Thanks all. Bye.
Thank you, everyone.
Bye.